Good afternoon, everyone. Welcome back to Virology. This is lecture number 2, and we're going to dive into, for the next 11 lectures, on what happens when viruses enter cells. So today we're going to talk about the infectious cycle. What is the infectious cycle? Well, it's everything that happens in a cell once a virus gets into it. Now, virologists usually divide the infectious cycle into steps to help us to study them. Of course, in reality, no such artificial boundaries occur. And what are those steps? Well, we talk about attachment and entry. We talk about translation. We talk about genome replication, particle assembly, and release. And these get their own lectures in this course, actually. Before we go any further, some important definitions. Now, when I talk about a susceptible cell, all that means is that that cell has a functional receptor for a virus. In other words, there's typically a membrane moiety, a protein or glycoprotein or glycolipid to which the virus attaches. So susceptible just means that there is such a receptor on the cell. It doesn't mean that the cell can support replication. So once the genome gets in, it might not replicate. So susceptibility only deals with the ability to bind to a cell. And a resistant cell has no receptor. It may or may not be competent to support replication, but there's no receptor. A permissive cell has the capacity to replicate virus, but it may or may not be susceptible. So here's a cell without a receptor, yet we're showing it as permissive, which means that if we put a receptor on those cells, the whole replication cycle could occur. And finally, a susceptible and permissive cell is the only cell that can take up a virus particle and replicate it. Now, although today we use animal cells in culture and plant cells and insect cells to study viruses, when viruses were first discovered, they couldn't be routinely propagated in cells. We didn't have it. We didn't know how to grow cells in culture. Most viruses were grown in laboratory animals or plants, as you would call tobacco mosaic virus. And we made do with that. Also, viruses could be grown in embryonated chicken eggs. And many different viruses, as shown here, could be inoculated into different parts of the chicken egg. Here's the embryo here. It's surrounding it is the amniotic cavity, and there's some viruses that will grow in the amniotic cavity in cells that are present there, of course. And then there's the allantoic cavity, which surrounds everything. And many viruses reproduce there as well. Now, influenza viruses are really the only ones that we still use eggs to grow in. In particular, the influenza vaccine strains are grown in massive quantities in eggs, as shown here. Why? Because it's efficient, it's cheap, and you get roughly 1 vaccine dose per egg. But we do grow influenza vaccines in other types of media as well. So we'll talk about that later. Now, the ability to study virus infections in cells wasn't possible before 1949, for animal viruses anyway. But in 1949, Anders, Weller, and Robbins found they could grow poliovirus in human cell cultures. These were primary cultures of embryonic tissues. And that gave them, all 3 of them, by the way, the Nobel Prize in 1954, and they made the cover of Time magazine. And that was an important discovery, of course. Then rapidly other viruses were shown to be able to be propagated in cells in culture. And today it's routine. We often use different kinds of cells to propagate viruses. And the three different kinds are shown here. We have primary human foreskin fibroblasts. These are just a subset of all the many different kinds of cells that we use. This is a primary cell line, which means you take the tissue, in this case foreskins, and you cut it up and mince it and digest it into single cells and plate them out on a plastic dish. And they form a monolayer, and these can be infected. Now, why foreskins? Hospitals make foreskins every day, right? From male births. You can get them if you are doing research. And many viruses grow in them, and they are normal cells. They die after about a month because they're not immortal. The next two virus cell cultures shown here are immortal, or continuous cell lines. These are mouse fibroblasts. It's called 3T3. And these are human epithelial cells, HeLa cells. So continuous means they grow forever. They won't die. You can grow them, you could freeze them, you can thaw them. In fact, these cells were isolated in 1951, and they still exist today. I'll tell you more about them. But these continuous cell lines are abnormal. They don't have the right number of chromosomes. They're transformed, that is, they're tumorigenic. And what that means is, as we will talk about later. We do have normal cell lines, normal in the sense that they have the right number of chromosomes, like WY38, human embryonic lung. And some of those can be used to grow vaccines. We can't use transformed cell lines to grow vaccines because there's the potential to have oncogenic DNA in them. Now, in my laboratory, we grew many different types of cells. On the left, you can see them grown in different formats like flasks and plates of different sizes, multi-well plates. But HeLa cells on the right are special in that they can grow in suspension. In other words, they will grow in liquid, not attached to plastic as on the left. There's a bar suspended in the jar here, or flask, with a piece of metal in it, and you put it on a stir plate and it spins, and it keeps the cells suspended, and they grow to very high density. So if you need a lot of cells, HeLa cells are very good. Now, HeLa cells, of course, were isolated from Henrietta Lacks in the 1950s. This young lady had a cervical cancer. During the examination, the doctors removed a piece of the cancer and brought it into the lab and found that actually it kept growing and growing and growing, and nobody had done that before. And in fact, she died shortly thereafter and never learned that. And then her family didn't know about it till many years later. They were kind of freaked out to think that their mother's cells were still growing. And all that story is described in this book by Rebecca Skloot, which is really good. You should read it. They cite me a few times. She had sent me the manuscript, for example, before publication, and I made some corrections that were really important. Okay, here is a quiz. Now, you can't use Socrative because I'm not online right now, but I'll tell you the right answer at the end. So a blank and blank cell is the only cell that can take up a virus particle and replicate it. So fill in the blanks. Naive and resistant, primary and permissive, susceptible and permissive, susceptible and naive, continuous and immortal. What were the two phrases that I taught you? Susceptible and permissive. A susceptible and permissive cell is the only cell that can take up a virus particle and replicate it. None of these other words are in the right order or are correct. Now, when you infect cells with viruses, in many cases, as the virus reproduces, the cells change. And the changes that happen, we call cytopathic effect, CPE. And here's an example. These are monolayers of cells. We're infecting them with poliovirus. And here's the uninfected monolayer on the top left. And then on the top right, 4 hours later, you can see the cells are rounding up. 8 hours later, most of them have detached, and by 12 hours, most of the cells are gone. So that's cell lysis. That's a particular kind of cytopathic effect. And this is a good time to show how this assay, CPE, was used to identify the SARS-CoV-2 in early 2020. So remember, late 2019, there's an epidemic of acute respiratory syndrome in humans in Wuhan. So what they did was they took bronchoalveolar lavages from some of these patients. So they took BALF, so they put a little saline down into the bronchial section of the lung from one patient, and they put it on cells in culture, Vero and HUH7 cells. And they say clear CPE was observed in cells after 3 days. And so here on the left are the uninfected cells, and here on the right after 3 days. And you can see clear cytopathic effect. So what I'm teaching you has real world application. And they say, there's a virus in there. And of course, subsequently, they tried to figure out what it was, and we'll keep tracking that in these early lectures. In fact, in this course, this was all happening in real time as I was teaching this course. Another kind of cytopathic effect is the formation of Syncytia. What is a Syncytia? Syncytia is nothing more than the fusion of multiple cells to get a giant cell with many nuclei. And many viruses can induce this process. For example, many viruses display on the cell surface this red Y is the glycoprotein, which can bind to the receptor on a neighboring cell, and that glycoprotein has fusogenic activity, so the cells fuse. And that can happen over and over to get these giant multinucleated cells, which are called syncytia. And here's a picture of one in culture. You see single cells and then giant cells like this one. This is typical of paramyxoviruses and other viruses like measles virus. There are many kinds of cytopathic effects. You don't have to learn them. You just need to know that they happen when you infect cells with viruses. And we talked about rounding up and detachment of cells. We talked about syncytium formation, many other kinds as well. Cytopathic effects. Okay, next, how do you count the number of viruses in a sample? So here on the right is a picture from my laboratory days. Each of these tubes has virus in it. How do you measure the amount of virus? So there are a couple of assays that you can do. They fall into two categories: infectivity assays or physical assays that measure virus particles and their components. Now, the infectivity assay was devised in the 1930s to study how bacteriophages multiply in cells. Remember, bacteriophages are viruses that infect bacteria. And what you can see here is that they have mixed a dilution of virus with host, typically E. coli in this case, and put it in the incubator. And the E. coli forms a lawn that you can see. And then when there is an infection initiated by a single particle, the result is a clearing or what was called a plaque. And you could count these plaques and express infectivity in terms of plaque-forming units per milliliter. This was adapted to animal viruses in 1952 by Renato Del Becco. Here's the paper. Production of plaques in monolayer tissue cultures by single particles of an animal virus. Obviously, this is a quantitative method. You can not only count viruses, but isolate the progeny of single particles and so forth. So it was very important to develop this. And here's how the plaque assay works. So let's say I give you a tube of virus, and I say, tell me how much virus is in this tube. So what you do is you go back to the lab, You make tenfold dilutions in medium or PBS. You take 0.1 ml of the stock, you put it in 0.9 ml. That's the 10 to the minus 1 dilution. You do it again, it's minus 2. And you go, it depends how well the virus grows. Usually you have some indication of that. And then you take samples of each dilution and plate them on cells. Now you have cells growing in the lab as a monolayer covering the plastic dish. You remove the medium and you add 0.1 mLs of virus. So the 0.1 ml of virus is essentially another tenfold dilution. Okay, so you then carry out a plaque assay. You add a semi-solid overlay composed of medium plus agar. You let it solidify and you incubate it. And then you can remove the agar and stain the monolayers and count the plaques. And you look for a plate that has a good number of plaques to count, not too many, not too few. It's like Goldilocks, right? Just the right number. And this one is good, 17. So that means here we have a dilution of 10 to the minus 6, 10 to the minus 7. So that would be 17 times 10 to the 7th. Figuring the dilutions. Then if you move the decimal point 1 over, it becomes 10 to the 8th. So that's the titer of the virus in PFU per mil. Very straightforward. Now here's an example of what's going on in these monolayers. On the top, you can see, let's say a single virus infects one cell. That cell is killed and it releases virus, which infects neighboring cells. So the clearing gets bigger and bigger. And we put an agar overlay on the cells to restrict virus diffusion to areas near the plaques, right? You don't want the virus to come out of these cells and spread throughout the culture, then you couldn't count the plaques. So that's the function of the agar overlay. And here are two microscopic views of plaques. Here on the left, you can see the cells in the center of this plaque, which are rounded up and detached. And when you remove the overlay, these cells are washed away. And on the right, some viruses don't form plaques, but you can put a gene in the virus that makes for a color, and the color tells you the plaque, and you can count them as well. Now here is the greatest movie ever made. It's a development of a plaque. What was done here was to infect cells with vaccinia virus, and then very early after infection, to identify what looked like a plaque, and then to do time-lapse photography, a frame every so often. And so this is, 4 or 5, 6 hours and so forth. And you can see the plaque growing as the cells are dying, and it gets bigger and bigger. And eventually, you have a big enough area to be a hole in the monolayer, and then you will remove the overlay and stain the monolayer so you can see the plaques. We used to do a lot of plaque assays in my laboratory. In fact, I used to have in my office what I called the wall of polio. These were thousands, 1,700 6-well plates where we had done a massive poliovirus plaque assay. And this was one of the virology classes of a couple of years ago, where students used to like to come and get their picture taken in front of this wall of polio. Can't do it anymore because it's gone. But it was pretty cool, I thought. Yeah. Okay, here's another quiz. When doing a plaque assay, what's the purpose of adding a semi-solid agar overlay on the monolayer of infected cells? Why do we add an agar? To stabilize progeny virus particles, to ensure that cells remain susceptible and permissive, to act as a pH indicator, to keep cells adherent to the plate during incubation, to restrict viral diffusion after lysis of infected cells. Of course, the answer is E. the presence of an agar overlay restricts diffusion of the virus as the viruses kill cells and move away from that first cell. And we can go back and see here, that's illustrated here. Only the neighboring cells are lysed because of the agar overlay restricting virus movement. And by the way, here's an example of a plaque assay. This is, I think, influenza virus, showing you that you remove this agar overlay and then you add a stain to the monolayer. And in this case, we used a stain called crystal violet, which stains living cells. And so the plaque, of course, has no cells, no living cells in it. And so it doesn't pick up stain. And so that's how you define a plaque. And so you can count them and determine PFU per mil. But how many viruses do you need to form a plaque? I've given you the assumption that you need just one, but how do we know? Well, in fact, that's one of the things that Delbecco asked in his first paper on the plaque assay. And it's a very simple question to answer. You can simply make dilutions of viruses, not tenfold, but twofold dilutions, and then do a plaque assay with each one. And so if only one virus particle is needed to form a plaque, that will be called one-hit kinetics. You get a line, you get a straight line. The number of plaques is proportional to the first power of the virus inoculated. If you double the concentration, you double the number of plaques. If you needed two plaques, you needed two virus particles to form a plaque, then you would get a curve like this. And actually, there are some viruses that require infection with more than one particle to initiate infection. But most animal viruses follow one-hit kinetics, so one particle is enough to start an infection and give you a plaque. Now, plaque purification is also commonly used to make virus stocks. So you do a plaque assay, you take the agar over the plaque with a plastic pipette, you put it in a little bit of buffer, the virus will come out of the agar, and then you can do another plaque assay. You can do this 3 times to ensure that you're working with just one virus. Now, some viruses don't form plaques, but they do kill cells, so we have ways to measure infectivity with those viruses. And one of these assays is called Endpoint dilution. And so what we do here is we take a 96-well plate. All right, so this is just a plastic dish with many wells in it that you can grow cells in. And then you infect each row with a different dilution of virus, -2, -3, -4, etc. And you incubate the plate for a few days, and then you count the number of wells that show cytopathic effect. So you can see at a low dilution, -2 and -3, all the wells are infected. And then you see at -4, one well is not infected. There's no CPE. And then at -5, you see 1, 2, 3, 4, 5 wells are uninfected and 5 are infected. So that is what we call the 50% endpoint. And that's a typical way to quantify in these kinds of assays. How much virus do you need to, in this case, cause CPE in 50% of the cells? And if you do this in tissue culture, we call it a TCID50. And so here the TCID50 would be 10 to the -5. All right, now, a little bit of a complication in virology is that not all virus particles are infectious. And that varies according to the virus. And we use this parameter to measure this, the particle to PFU ratio. And that simply is for any given virus, if you take the number of physical particles, which you have to find a way to count them, and divide by the number of infectious particles, that's a measure of how many virus particles are infectious. And so the best on this table is Semliki forest virus, for which almost every particle is infectious. So the particle to PFU ratio is between 1 and 2. But you can see for other viruses, it varies widely. For polio, it's between 30 and 1,000, and for papillomaviruses, it's 10,000. So only 1 in 10,000 particles is infectious. That's crazy, right? It can complicate experiments because you don't know the effects that you observe if they have anything to do with infectious particles or not. Anyway, why is this so? Why is this so? Now, I already told you that a single particle can make a plaque, right? But not everyone does, right? As you can see by the particle to PFU ratio. Sometimes the particles are damaged here. You can see in this electron micrographs, these are polioviruses, and some of them are empty. They have no RNA in them. Some genomes may have lethal mutations. You know, the infection cycle, as we're going to see, is pretty complicated. So a failure at any step can nix the whole cycle. So it's not surprising. Why some viruses are better than others is just not clear. Nobody really understands. Okay, in the particle to PFU ratio, particle can best be described as one of the proteins which makes up the virus, a virus which may or may not be infectious, a virus which is infectious, a virus which is not infectious, elementary or composite. The particle to PFU ratio. So remember, the particle is everything in the PFU or the plaque forming unit. So the answer is B, a virus which may or may not be infectious. Another important tool for studying viruses is the one-step growth cycle. We've touched very briefly on this, but we'll go a little bit into more depth. This was developed in 1939, studying bacteriophages of E. coli. And in this experiment, what you do is you allow the viruses to absorb or attach to cells. You then dilute the culture so no more attachment occurs. You incubate the infected cells, and then you take a sample at different times after infection, and you measure virus by plaque assay. Very simple assay. And here's what you get. You have on this graph on the left, number of infectious particles, PFU per mil, versus time on the x-axis. When you infect cells, and then you dilute them, for a while, there's no evidence of new virus production. And then suddenly, the time varies. It's called the eclipse period because infectivity is eclipsed. Suddenly, you get a burst of virus production, which plateaus as all the cells are killed. So, remember, this is what distinguished viruses from bacteria. And we know now in the eclipse period, that's the time when the parts of viruses are being made and assembled into virus particles. That's a one-step growth curve, but you can do multi-step growth curves where you infect with less virus, and then you'll have multiple bursts. You'll have your first eclipse, your first burst, but not all the cells are infected because you've added a small amount of virus. The virus produced in the first burst will then go on and infect other cells, and you'll get a second burst. Sometimes you want to do a multiple-step growth curve depending on what you want to learn from the experiment. You can do these with animal cells and their viruses as well. Here's an example with adenovirus, a large DNA virus. Again, PFU per mil on the y-axis, hours after adsorption on the x-axis. We have an eclipse period again, as usual. And here you can see it takes 12 hours before you start to detect viruses in the cells. So the eclipse period, we see no infectivity in the cells. And then there's a new period here, the latent period, where the new virus particles can only be found inside cells. And then starting at 16 hours or so after infection, you can see the release of virus particles from cells. And that defines the latent period. So you can see some viruses, it takes some time between when they are made in the cell and when they can get out of the cell. All right, contrast this, remember, with bacteria. This is really important to remember. In bacteria, when you put a single bacterium into a broth culture, there's no eclipse period like there is in eukaryotic viruses, or any virus. The bacteria pretty much start to divide. There may be a slight lag, but we do not define an eclipse period in bacteria. Bacteria start to divide. 1 becomes 2, 4, 8, so forth. They don't make the parts first and assemble bacteria. The bacteria simply split by binary fission. Now, a key to the one-step growth cycle is what we call synchronous infection. We have to get all the cells infected. How do we know if all the cells are infected? Well, there is a way for us to know, and that's called the multiplicity of infection, or MOI. MOI is simply the number of infectious particles added per cell. Or, put another way, the amount of virus in PFU divided by the number of cells. It is not the number of infectious virus particles that each cell receives. So, for example, if you add 10 to the 7th virus particles to 10 to the 6th cells, that gives you an MOI of 10, but each cell does not get 10 particles. Why not? Well, infection is a random process. It depends on the random collision of virus particles in cells. When you mix cells with virus, some cells are uninfected, some get 1 particle, some get 2 or 3 or more. And I like to describe this in this way. You have a bunch of buckets here, and then you have a box of tennis balls, and you throw the tennis balls at the buckets. Each bucket's not going to get the same number of tennis balls. Some will get none, some will get 1, 2, 3. And there is a mathematical formula that describes how many virus particles or even tennis balls you would get per bucket you get depending on how many you add. So if you have 1 ball in here versus 100. So the Poisson distribution is a mathematical equation that can tell you how many virus particles each cell would get depending on how many you add. And this is the Poisson distribution equation. This is a graph of the function showing you the events that occur at different inputs, where PK is a fraction of cells infected by K virus particle, and M is the multiplicity of infection. Now, don't be daunted by this formula. It can be simplified. And so if you want to know the number of uninfected cells, that would be P0. That reduces to e to the minus m, e being the natural log. So you just plug in your MOI there. If you want to know cells receiving 1 particle, that's P1 equals ME minus M. And then if you want to know the cells that are multiply infected, more than 1, This is the formula for that, which is, it's obtained by subtracting from 1, which is the sum of all probabilities for any value of k. The probability is P0 and P1. You can see E minus M and ME minus M in there. So let's look at some examples of this. So if you take 1 million cells and infect at an MOI of 10, which means you're adding 10 to the 7th PFU, 45 cells are uninfected, 450 get 1 particle, and the rest get more than 1. So an MOI of 10 is a really good way to infect every cell, right? If you infect 1 million cells at an MOI of 1, now you have 37% uninfected, 37% get 1 particle, and 26% get more than 1 particle. So now you're not going to have a single-step growth curve. And if you do an MOI of 0.001, most of the cells are uninfected. But some are, and eventually they will start reproducing. So that's MOI. If cells are infected at an MOI of 10 in a 1-step growth cycle experiment, in the growth curve, you will likely see multiple bursts of virus release, multiple eclipse periods, a single burst of virus released, no burst of virus release, or asynchronous infection. Now remember, what does MOI 10 give you? As I just showed you, MOI of 10 infects most of the cells. So you're going to get a single burst of virus relief. It's going to be a 1-step infection. All right, so those are ways of measuring infectious particles. It doesn't always work for all viruses, and for that we have developed many ways to measure particles, and these include hemagglutination, an assay where red blood cells stick together because of the presence of virus particles, electron microscopy in a pinch. Some virus particles have enzymes in them. We can measure those. And then we can use serology, where, for example, antibodies can be used to detect viral proteins or viral antibodies. And finally, nucleic acids, of course, where we simply sequence a sample and that can tell you roughly how much virus particles are present. So let's go through some of these. Hemagglutination is very interesting. It depends on viruses that bind to sialic acid. Sialic acids are receptors for some viruses. These are sugars on the surface of cells. They're present on red blood cells. And if virus particles will bind the red blood cells, then the red blood cells will form aggregates, and you can visually see these aggregates. So here at the top, for example, is an example of doing a hemagglutination assay with influenza virus. So we have dilutions of virus, and then we incubate and look for the red blood cells coating the well. So if they form this lattice, they will coat the well. If there's no virus present, then the red blood cells fall to the bottom of the well and make a nice little button. So you can see the top sample doesn't have any virus in it, but the bottom sample will hemagglutinate out to 1 in 512. So that's the HA titer, 512. It's a good way of quickly measuring the presence of virus because it can be done in about 30 minutes. You can measure enzyme activity. Retroviruses, of course, famously have reverse transcriptase enzyme in them, as well as other enzymes. But reverse transcriptase can be assayed readily. And you can see that here. These are cells infected with either mock-infected or with a retrovirus and a 1 to 10 dilution. These are supernatants, basically, of infected or uninfected cells. And the reverse transcriptase is detected by incorporation into a DNA product. So that's very easy to measure. Now, there are serological methods, right, which are very powerful and important. These are called ELISAs, enzyme-linked immunosorbent assays. We can detect either viral antigens or antibodies. So on the left, we have an assay for viral antigens, right? So there's the viral antigen in purple. And what you do is you take an antibody to the viral antigen and you stick it onto a plastic support. This is typically done in a 96-well format. And then if you have a clinical specimen, you can flow it over. And if there's viral proteins present, they will stick to the antibody. And then you add a second antibody that binds a different epitope on the viral protein, which has an indicator like fluorescence or something else that you can easily visualize. So this is the first antibody is called the capture antibody. And the second antibody is the second antibody, which is used for detection. So again, that's looking for viral proteins. On the right, we're looking for viral antibodies. So in this case, you put the viral antigen on the plastic. And then you can put your sample on the plastic. If there are antibodies to the virus in the sample, they will stick to the antigen. And then you use a second antibody with an indicator molecule. So these are two different ways of knowing if someone has been infected. On the left, if someone is currently infected or very recently. And on the right, of course, you have antibodies to an infection that persists for a while. So it can tell you have a history of infection with that virus. Now, this immunofluorescence was very important for identifying SARS-CoV-2. So remember, I showed you a picture of the CPE produced after putting the BALF fluid from a pneumonia patient onto cells. And we got CPE. So they said, that's good, there's a virus. Well, what virus is it? Well, they suspected it might be a coronavirus because, in 2002, SARS emerged from bats and caused a small global outbreak. And so they had reagents to coronaviruses and they had an antibody to the viral nucleoprotein, which is rather conserved among these viruses. And so they used that to stain those infected cells. So on the left are the control cells that are uninfected, and on the right are the cells where the BALF was added. And you can see the red. That shows you there are viral proteins made. So this experiment shows you that the pneumonia patient was infected with a coronavirus of some kind. So this is a very powerful technique. Now, if you've ever taken a rapid COVID antigen test, a RAT, this is how it works. In fact, I just took one the other day and it was negative. So what you do is you have this format for the test. It varies depending on the company, but it's essentially a pad on a support with different zones. And you take your clinical sample with your antigen, which I've shown here in brown, and the actual test material has here in the very beginning monoclonal antibodies to the antigen that you're looking for, linked to colloidal gold. And then you have two other zones where you have antibodies to the antigen. And then the final one, that's the test line. That'll be positive if you're infected. And then the control line, just to see if the assay works, these are antibodies to the antibody. just to show that the whole thing is working, that something is flowing down and binding. So you put your sample at the end, it begins to flow down this pad by capillary action. If there are antigens present, these antibodies will pick them up and they will move down. So here now we have antibodies bound to the antigen. And then these antibodies will bind to that first row of monoclonal antibodies via the antigen. You can see, and that will give you your positive result or negative if there's no antigen. But if there's no antigen, the antibodies will continue to flow and they will reach the control line. They will bind to the antibody and give you that positive, showing you that the assay worked. You will get that line whether or not there are antigens present, right? Because it doesn't matter, because the antibody is binding to the Fc portion of the antibody. So that's how rats work in general. A couple of other things we use in virology quite a bit. One is green fluorescent protein. As you know, first worked at Columbia years ago by Marty Chalfie. It's a protein made by jellyfish that glows green under fluorescent light. And so you can actually make variants in different colors, or you can isolate variants of different colors from different species. But you can incorporate the gene for this protein into viral genomes. So here's an example of where the gene was, for GFP, was incorporated into the HIV-1 particles. And you can actually see the virus particles in green. So this is a light micrograph, and you can't see virus particles for the most part by light microscopy, but in fluorescence, the light thrown out amplifies the size of the particle so that you can see it. And what's happening here, these are microtubules stained in red, and the virus particles are moving towards the nucleus on microtubules. Sequencing of nucleic acids has been also important. This allows us to do metagenomics, just sequence everything in a sample, whether it's soil or water or a clinical specimen. Can identify new pathogens. Eventually, this was done to identify SARS-CoV-2. Of course, the human genome was done years ago, the first human genome. It took 10 years at a cost of $3 billion. But technology has improved so that today you could have your genome sequenced and it would only cost $1,000. How about that? When I was a postdoc, this is how we did sequencing, by running these gels. It was very tedious. It took a long time. I sequenced the genome of poliovirus, which was 7,440 bases. It took me a year. You could do that in minutes nowadays because the new high-throughput sequencing methods are so powerful. It's amazing. The ability to sequence genomes, and not just sequence, but many genomes, has enabled us to construct phylogenetic trees. And we'll talk about these from time to time in this course, so I want to explain now what they are. A phylogenetic tree measures the genetic distance between genomes and basically organizes viruses according to the closest sequence. So here we have 10 virus sequences, and we use a computer algorithm, and we compare the 10 sequences in pairwise comparisons, and we display them as who is the closest. So here, virus 1 and 2 are the closest, 3 and 4, and obviously 3 and 4 and 1 or 2 are very close, and more distal from 9 and 10. So each division is called a node, and eventually they all derive from some ancestor, but often we don't have that ancestor. So the this blue box is called the presumed ancestor or the root of the tree. And if you go left to right, you're measuring basically the degree of genetic change, where the scale, and here's the scale for this particular phylogenetic tree, tells you the changes per length of sequence, so you can tell how distant these viruses are. And then this other number here is the measure of support, telling you to what extent sequences to the right of this node cluster together better than other sequences. So this was used early in the SARS-CoV-2 outbreak. Remember, we had isolated virus from one patient in cell culture, and so that virus was taken and its genome sequence was done. And in this paper, they constructed a phylogenetic tree of genome sequences of all the known coronaviruses up to that point. And so you can see here, these red sequences are from the 2019 samples. They all cluster very closely together, and the human SARS sequences from 2002 are quite distant. So this was not SARS-1. It clearly showed you that. And it was distanced from most of these known coronaviruses, including human common cold coronaviruses and a variety of animal coronaviruses. The closest relative to this virus was a virus called bat coronavirus RATG13. And in fact, this doesn't exist as a virus. It just existed as a sequence. And you may hear people saying that the lab, the Zhongli Shi lab working on this virus, transformed it into SARS-CoV-2. This is virtually impossible. This virus, even though it's the closest to SARS-CoV-2, is still 1,200 bases different. It would take forever for you to change it. How would you know what to change it to? And anyway, they could not recover virus from RATG13. So saying it's an ancestor is just nonsense. Reflects a lack of understanding of virology. Another essential technique for virology research is PCR, polymerase chain reaction. Huge impact on research, on industry, and diagnosis. So this comes from the study of bacteria that grow in hot springs. Remember last time we talked about the grasses that grow in hot springs? And studying these extreme environments can be very useful. So Thomas Brock, a microbiologist working in the 60s, wanted to know what the bacteria growing in these were like. And he isolated one strain called Thermus aquaticus. Eventually, people found that this bacteria has a DNA polymerase that can withstand high temperatures, as you might guess. And so that DNA polymerase was the foundation of PCR, because in PCR, you take your original DNA, you take a piece of DNA, and you want to amplify it because there's not enough there to see without amplifying it. So you start by denaturing the two strands at 100 degrees centigrade, basically. And then you add DNA primers, and then the DNA polymerase will extend those primers. So now you have two double-stranded copies where you had one before. You repeat that over and over. You have 4 and 8 and 16. So you need a polymerase that can withstand multiple shifts in temperature up to 100 degrees. And TAC polymerase, Thermus aquaticus polymerase, can do that. Very important technique for research, industry, and diagnosis. Now, in terms of virology, it's used often to measure nucleic acids, both DNA and RNA. We can convert the RNA to DNA and then do the PCR reaction. But a caution in that PCR product is not the same as infectious virus. Here's an experiment that shows that for Zika virus. So in this experiment, they infect mice with Zika virus, and then they measure infectious Zika virus and Zika virus RNA by PCR. You can see the mice shed in semen infectious virus up to about 21 days, but nothing after that. Yet Zika RNA persists for over 60 days. But obviously, there's no infectivity at that point. So PCR product is not infectious virus. And the control experiment here is if you vasectomize the mice, that dramatically reduces shedding of infectious virus. And for many RNA viruses, you can detect RNA long after the disappearance of infectious virus, including SARS-CoV-2. Surprise. Remember early in the COVID pandemic, if you were PCR positive, you had to stay home. How did we know that you were actually infectious? Well, we didn't. Eventually, people did experiments to sort that out. We do have a plaque assay for SARS-CoV-2, but nobody did them. A clinical assay, a plaque assay is difficult. Hard to automate and hard to do a lot of assays at once. But here's an experiment showing you that, if you're infected with SARS-CoV-2, you shed infectious virus here in blue for a certain number of days, limited, say 10, 12 days. But you continue to shed SARS-CoV-2 RNA here for up to 22 days and sometimes longer. So a lot of people were made to stay home when they didn't need to. So it was hard to answer these questions early in a pandemic where everyone is panicking, but sometimes people didn't even consider it. And so that was wrong. It's a difficult question because the sample you get from every person is slightly different. So you'd have to have a way of normalizing, maybe to mitochondrial DNA or something like that, but that wasn't done. As you know, the rapid antigen tests were subsequently developed, which look for protein. And there's a little better congruence with infectivity when you look at protein. It's not perfect, and this is something we need to deal with in future pandemics. All right, that's a overview of what goes on in the infectious cycle and how we measure it. Next time, we're going to talk about the genomes of different viruses and how we can manipulate them to study them.